Good morning, grade 10. Today we just complete a small part of lesson 3 and we started reviewing for the second assessment. So at first let's review main concepts of fingerprinting, DNA fingerprinting, which is a technique that's used to identify people at molecular level. We said that the DNA fingerprinting is a representation of parts of individuals' DNA that can be used to identify a person at the molecular level. And DNA fingerprint is itself a type of restriction map, which we learned before, that we prepare it by isolating or extracting DNA from a sample. We take a sample. Let's say, for example, a sample from crime scene, blood sample, hair sample, skin parts, and they isolate the DNA or extract the DNA from it. Then the DNA should be cut by restriction enzymes, then be separated by the gel electrophoresis to DNA fragments. This is the restriction map. So DNA fingerprint is a type of restriction map, and it's based on parts of individuals' DNA that can be used for identification. But you should keep in your mind that it's based on non-coding regions of DNA. That means it doesn't base on the coding regions which have the genes for coding for proteins. No, there are parts of DNA that are non-coding regions. So the DNA fingerprinting based on non-coding regions which have repeating DNA sequences. Okay, so number of repeats differs between people, and these repeats form bands on the gel electrophoresis. This band pattern on the gel is the DNA fingerprint. As you can see here, this is a DNA sequence of 33 base pairs that can be repeated many times in a sample of a person DNA. This sequence may be found in many people, the same sequence, but the repeating of this sequence is different. So some persons which have this sequence have 4 repeats of it. Other person, as you see in person B, they have only 2 repeats of it. So this is what it differs in the fingerprint of DNA, the repeats, the number of repeats of the sequence, specific sequence of the DNA. Okay, so DNA fingerprinting is to identify people has become reliable and widely used process. Why? Because the specific nucleotide sequences that are repeated can be found in everyone. So these sequences can be found in everyone. Okay? But from one person to another, the number of repeat sequences can differ greatly, even among brothers and sisters. So DNA fingerprinting compares at least 5 regions of the genome. That's why it is more certain that the pattern of DNA fragments in the fingerprint is unique. Now, the more regions of DNA that are studied, the less likely it becomes that another person would have the same DNA. For this reason, DNA fingerprint is considered very reliable to identify persons. And to make it more clear, if you look at this restriction map of DNA fingerprinting, you will find here fragments of DNA at different locations. Okay, they should use at least 4 locations to compare the fragments, because it is impossible to find all the repeats of each fragment the same. You can find them the same at some locations, but it's impossible to find the repeats are the same in all the locations. Okay, now this is for this part. We also started reviewing for the exam, the second exam. We talk about replication. You should know that replication, it's a process of copying the genetic information for the cell, and it happened once in the cell cycle, where it needs the DNA polymerase to to make the copy. Okay, there is a complex enzymes that start the process of replication. First, it will unzip the double helix, and the DNA polymerase start bonding free-floating nucleotides to the to the template strand nucleotide. All right. Then they form covalent bonds between nucleotides as hydrogen bonds. Now we said the DNA replication is semi-conservative, which means that the two new molecules of DNA are formed, and each with an original strand and a newly formed strand. We also said that replication happened at many locations to make the replication happen quickly. And DNA polymerase, it has another function, which is correcting the errors in replication. Okay, see you. Take care.